Hi everyone, we're going to talk briefly about Chapter 41, Genetic Disorders. Genetics has been a very hot topic of late, and we are learning more about the human genome and how it transforms into understanding genetics, embryology, physiology, and disease processes. It's no longer the science of studying just a single gene, but how genes interact with the whole system and how they're influenced by other components of our lives, environment, events, epigenetic factors. So let's talk briefly about some basic principles of genetics, and this is going to just be brief. When one sperm and one egg come together, they create a unique DNA package for each of us, and then the whole total DNA package is called the genome. Within each cell, genetic information flows from the DNA to the RNA to protein, with each of these genes being coded with up to 20 different proteins. Each DNA is composed of a string of nucleotides, and they consist of deoxyribose phosphate groups and a base of either adenine, cysteine, guanine, or thymine. We know that each person has a unique DNA package that represents several million different possible combinations from 4 grandparents' genetic materials, but the base sequence is the same at approximately 99.9%. And those differences arise from only 0.1% of the DNA, so even the slightest alteration in the DNA can have some significant consequences, such as sickle cell anemia that's caused by an alteration of just a single nucleotide. With chromosomes, let's go to the next one. Chromosomes, yes. So cytogenetics is the study of genetics at the chromosomal level, and each chromosome has a strand of DNA. We know that the nucleus of all human cells, except for gametes, contain 46 chromosomes in 23 pairs. There are 22 pairs that are called autosomes, and they are the same in both males and females. And then the sex chromosomes make up the remaining pair, and they differ between males and females. And as we know, 2 X chromosomes for female and an X and a Y for male. These are numbered by size from 1 to 22, 1 being the largest to 22 being the smallest. And the picture of chromosomes lined up is called a karyotype. So human somatic cells are diploid with 23 paired chromosomes, and they replicate by the process of mitosis, where they create 2 identical daughter cells. And gametes are haploid with 23 chromosomes. They are produced in the ovary and testicle during meiosis, where there's the exchange of genetic material. And there's a random assortment of mom and dad genetic material in the daughter cells. When fertilization occurs, there is the fusion of those cells, and then the 46 chromosomes complement is restored. Chromosomes have both a long arm and a short arm, and an ideogram is used to show the size and the banding patterns. Bands also tell more about the location of the genes on the chromosome. Telomeres are the protective caps at the end of each chromosome, and they contain genetic information and protect it on the chromosome. So each time the cell divides, these become shorter and they lose their protective function over time. Stress can impact the telomeres, and increased risk for adult health problems may be related to stress and the shortening of these telomeres. So with genes, those offer the specific coding sequence of the DNA. There's 64 different codons in genetic codes, and then 1 gene codes for the average of 3 proteins. The genotype is the individual's collection of genes. The phenotype is what the individual's genotype can do, because gene expression is both regulated by molecular mechanisms and it can be impacted by environmental factors. So we know that 2 copies of each gene are inherited, 1 from each parent, and homozygous are 2 alleles that are similar, and heterozygous are 2 that are different. So humans may have anywhere from 20 to 25,000 genes, and each cell contains a gene, although all are not active at the same time. And the mechanisms to activate genes can be turned on and off by the body. Exons are the coding sequence, and introns are non-coding sequences, and they are located on the chromosomes. So we talked previously about an allele, and these are regarded as a mutation when a genetic variation is found in less than 1% of the population, and these are often disease-causing, or a polymorphism when it's found in greater than 1% of the population, and these are usually considered normal variations. There are single nucleotide polymorphisms, SNPs, which are single-based alterations common, and they're not directly related to disease, but they can help identify those that are at risk for a multifactorial disease or an adverse response. Mutations can directly cause disorders, and they can be subclassified as point mutations, nucleotide, repeat expansions, copy number variants, or chromosome mutations. So let's talk about those just for a moment. Point mutations are single base pair changes, and they occur at the level of the nucleotide, and they're capable of changing the function of the gene or the gene product. They're responsible for many single gene genetic disorders such as cystic fibrosis and sickle cell. The nucleotide repeat expansions can occur beyond the single point changes, and it's a repeat expansion. A repeat expansion is a mutation that increases in number of times the short DNA sequence is normally repeated. And when the number of repeats is high enough, then a genetic disorder may result. Copy number variation involves a large area of chromosomes beyond point mutations and repeat expansions. When the egg and the sperm unite, there's an unequal crossover throughout the genome, and when this happens, children may have lost or gained additional copies of genetic information that were present in either of their parents' chromosomes. And this is often seen in common psychobehavioral diseases, such as mental retardation, autism, and schizophrenia. And then chromosome mutations, these are seen in larger segments of the chromosome that are deleted, duplicated, rearranged, or translocated. These mutations usually result in multi-organ large effects, such as Downs, in which the individual can have neurological issues, eye, ear, orthopedic, cardiac, and other abnormalities. Next, let's talk about types of genetic disorders. There's monogenetic disorders, and this happens when a mutation affects just a single gene. Dominant, meaning a mutation on one of the alleles, recessive on both, or X-linked confined to just the X chromosome. In chromosome disorders, there can be changes in the entire structure of the chromosome or in the number. So the Prader-Willi is an absence of a group of genes, and Down syndrome is an extra copy of the chromosomes. Mosaicism is altered chromosomal arrangements in some cells but not others, and with this disorder you often see milder symptoms with a better prognosis. Multifactorial disorders are a combination of genetic and environmental factors such as spina bifida. They tend to cluster in families. The exact risk for occurrence is difficult to predict, even though we know that they cluster in families, and it's difficult because of the individual or couple's precise genetic environmental risk are often unknown. So you may see a population-based recurrence risk rather than a personal recurrence risk. Teratogens are any agent that results in or increases the incidence of a congenital malformation. Teratogens have traditionally been considered an environmental toxin, altering embryonic and fetal development. but it is thought now that there can be genomic factors that can modify the effects of the teratogen. So the same teratogenic exposure to one embryo may not cause the same effect to the other embryo. The majority of the individual's DNA is found in chromosomes within the nucleus of the cell. A small amount of genetic material is found in the mitochondria, in the cytoplasm outside of the nucleus, and this genetic material is known as mitochondrial DNA and contains 37 genes. And these disorders are typically progressive, affecting the brain and the muscles, and they're exclusively transmitted on the maternal side. Let's next talk a little bit about patterns of inheritance. So on page 1153, this is the table that we see before us. We talk about different gene expression variables, penetrance, expressivity, pleiotropy, variable age of onset, etc. It gives definitions and then examples of the different variables. So we're not going to go over those, but I encourage you to review this table. There are different patterns of inheritance, and we know that gene disorders are caused by a mutation in a single gene, are usually inherited in a couple of different ways, and these are known as the Mendelian inheritance pattern. And there's an autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. We're going to go over those. Autosomal dominant is an inheritance of a single copy of a mutated gene, and we know that it's passed from one parent, but results in genetic disorders because it is gene dominant. So the parent that it has come from has the disorder, and there's the risk of inheriting it is 50% for each child, regardless of the sex and independent of having an affected sibling. So it's important for clinicians to review a child's family health history and consider autosomal dominant inheritance when certain conditions are discussed. Autosomal recessive requires inheritance of 2 copies of the mutated gene, one from each parent. And so offsprings with only 1 copy are considered carriers, and carriers may pass the genes to their children but are themselves unaffected. So the risk for inheriting is 25% for each child, and a 50% risk for being a carrier and 25% risk of being unaffected. Examples of autosomal recessive disorders include cystic fibrosis, thalassemia, sickle cell anemia. X-linked dominant are single abnormal genes on the X chromosome. If the father is affected and the mother is not, all female offsprings will inherit the disease. If the mother is affected and the father is not, 50% chance that each daughter or son will inherit this. Some examples of X-linked dominant inheritance include Rett syndrome and vitamin D resistant rickets. X-linked recessive usually occurs in males and is seen when males have only 1 X chromosome, so a single recessive gene on that X chromosome will cause the disease. Examples of X-linked recessive disorders include hemophilia A and B, G6PD deficiencies, and Duchenne muscular dystrophy. Non-traditional inheritance patterns include your mitochondrial inheritance, and this is passed by mom as mitochondrial mutations, as only eggs contribute to the mitochondria in the developing embryo. Examples of this genetic disorder include myoclonic epilepsy. On codominant inheritance, it's a disorder as having a codominant with 2 different alleles for a gene can be expressed, and different combinations result in slightly different proteins. An example of this is seen in blood type determinations, your your ABO blood grouping. Next is your genetic imprinting, and this occurs during embryogenesis, where one gene copy is activated and another is inactivated. So if there's been improper imprinting, this can result in two active or inactive copies of a gene. An example of this would be Angelman syndrome. And then uniparental disomy, are two copies of chromosomes from one parent and none from the other. Often this does not have an effect on the child's health or development because most genes are not imprinted. This may or may not cause any diseases, depending on the specifics. Next, let's talk about epigenetics, and this is the study of changes in the gene function that can occur without a change in the DNA sequence. Epigenetics regulates which genes get turned on and off. It's impacted by chemical exposure, diet, endocrine disruptors, maternal age. Unlike the genome, the epigenome is modifiable. Most common adult disorders are now believed to be caused by epigenetic changes that occur during the individual's childhood, and these can include heart disease, hypertension, and obesity. So let's see how genetic implications can impact pediatric care. We know that when we're going in, we need to get a good assessment. We need to get a detailed family health history. We need to recognize if there's any genetic red flags. Emphasis is placed on understanding genetic screening and working with children and families to understand these implications. A complete head-to-toe physical and a good developmental assessment, and then a comprehensive family health history. It's important in primary care to take the time to collect the child's family health history and pedigree. You have probably seen in practice a 3-generation pedigree, and it's a visual record of genetic links and health-related information. On page 1158 in the book, table 41-2, there's some good information on general screening questions for genetic conditions when you're trying to obtain a good history. Table 41-3 gives some definitions for the degree of relationships between individual family members and their shared genetic material. And I believe this is on this slide. Got a little bit ahead of myself. And so let's talk about some genetic red flags that may come up from your history taking. These are things that indicate a potential for a genetic risk. There is the rule of two, TOO and TWO, too many of something, or two people or events that occur. There is also another mnemonic called the screen mnemonic, and it's found in your book on 1159 for table 41-4. That's good to help to remember the important components to look for and ask for when getting a family health history. So what you're looking for is multiple affected members with the same disorder, earlier age of onset than is expected for the disorder, condition or disorders in less often affected sex, appearance of the disease and absence of known risk factors, ethnicity and ancestral background, unusual close relationships, multifactorial or bilateral occurrence impaired organs, and intellectual impairment. Physical findings indicating a genetic disorder, you're looking for abnormalities such as cafe au lait spots, growth problems, congenital anomalies. You may be looking for a neurological anomaly, hearing or vision loss, developmental delay, mental retardation, seizure disorders. Physical findings indicating genetic disorders may be major or minor. Would be considered major, or two minor abnormalities. You may want to consider a genetic etiology. You're looking for malformations, birth defects, deformities, disruptions, dysplasia, syndromes, and associations. Screenings may be used if there is asymptomatic populations to identify those that may need further evaluation. We know that there's newborn screenings which identify disorders that can benefit from early diagnosis and treatment. There's also the National Recommended Universal Screening Panel, the RUSP, which is the 31 core and 26 secondary conditions that are screened for. There's prenatal screening, which is used to detect genetic or congenital disorders before birth. We have diagnostic genetic testing, which is used to confirm a diagnosis. And the main types of testing for this would be karyotype, FISH, biochemical testing, chromosomal macroarray molecular testing, and next generation sequencing. So in more detail on those, karyotype is identifying and evaluating the size, shape, and the number of the chromosomes. FISH is locating and detecting specific areas of a particular chromosome. Biochemical testing is studying the amount, the activity level, and the structure of the enzymes and proteins. A chromosomal microarray is detecting microdeletions and duplications. Molecular testing is detecting specific single gel mutations, and next-gen sequencing is detecting a single mutation among many genes. We can also do carrier testing, which identifies individuals who have one copy of a gene, and it's usually used with family planning. And other testing is predictive or pre-symptomatic tests for asymptomatic individuals. We've heard of pharmacological testing to learn which dose or drug is best for an individual, and forensic testing to establish biological relationships. When a family history is positive, then there needs to be a genetic referral. If you have children with developmental growth or structural disorders, they need to be referred. And genetic counseling is so important and needs to be a specific aspect of the referral. So children with a genetic disorder, primary care is the foundation. So having a good medical home, having age-appropriate health supervision guidelines, and then specific guidelines for children with certain conditions to help manage their primary care. And lastly, our ethical concerns. A few of these ethical issues includes the right to know or not know, the duty to warn, disclosing of incidental findings. In 2008, the Genetic Information Non-Discrimination Act was passed. The intent of this legislation was to protect individuals from misuse of genetic information to health insurance and employment, and remove barriers to the use of genetic services. Under this act, health insurers cannot use an individual's genetic information to set eligibility requirements, establish insurance premiums, or request certain genetic tests. So this is important information to pass along to families, and employers cannot request, require, or purchase genetic information about an employee or a family member, and they cannot use an individual's genetic information in decisions about job hiring, firing, assignments, or promotions. If you deal with families that are involved with genetic testing, then knowing more about these ethical issues may be able to assist you in answering those family or patients' questions. So I believe that is the end of chapter 41. Thank you.